row.
Okay, what we're going to do is we will spend the first 15, maybe 20 minutes wrapping up stuff for the exam on Thursday. And then I'm going to do some stuff to help get you ready for lab. That should make lab go a little bit faster and then maybe give you some extra time. You can start working on other things in lab besides the little project we're starting, which would include starting to get to work on your unknown. Okay. So what we're doing is we're talking about the last aspect of the second line of defense that we're going to cover. Remember, second line of defense is part of your innate immunity, something you are born with, as opposed to the third line of defense, which you acquire during your lifetime as a result of exposure to various things in the environment, antigens. So what we're talking about here is inflammation. And inflammation is a pretty complicated response to a wide variety of different types of injury. And that injury may or may not involve breakage of the skin. If it does involve breakage of the skin, then there's always a chance that with that first line of defense breached, that pathogens could be involved. But inflammation does not have to be associated with entrance of pathogens into your tissues. It can be a result of entirely mechanical injury, like a twisted ankle, for example. So in this particular example, we're showing an actual injury that involves breaching that first line of defense, your skin barrier. Now, there's some very complicated interactions that are taking place. And those interactions involve several players, one of which are your host tissue cells, which have become injured. Another player in the game are pathogens, if they're present, may or may not be. Another player or set of players in this process are various immune cells. The major immune cells are neutrophils and their cousin, the mast cells. Macrophages, to a lesser extent, lymphocytes, and then tying this all together, and a really important part of understanding inflammation, are a variety of cytokines. So there's a lot of chemical crosstalk and signaling that's taken place between host cells, pathogens, immune cells. So it gets a little complicated. We're going to look at it in a fairly superficial way, but hopefully to give you some kind of an idea of some of the interactions that take place, and particularly what are some of the major cytokines. Because ultimately what we would like to do is to develop an understanding that allows us to understand why do we get the particular symptoms that we get when an inflammatory response is initiated. And what are those symptoms? The classical symptoms of inflammation are the rubor, calor, tumor, dolor, redness, heat, swelling, and pain. Now, from just kind of an overall standpoint, what would you suppose was responsible for, well, let's step back for just a second. What were the goals of inflammation? Okay, so we want to have some kind of a response that could destroy pathogens and also repair damaged tissue. Okay, And what's going to do that? Well, a lot of your immune cells are going to be involved in that particular part of the response. Where do we find those immune cells typically? Well, we find them in the lymphatic system, and we also find them in the circulatory system. But does it do me much good if I've got a lot of immune cells in the circulatory system in my leg and the injury's in my finger? No, we've got to somehow bring that blood flow and the cells that it carries to that site of injury. And so that's one of the other goals of inflammation, is to divert blood flow containing these protective cells to the site of injury. And then once those blood cells are there, they can begin this process of killing pathogens, starting to repair the tissues. And then we said the third function was to kind of cause you to back off a little bit. Okay, that pain in particularly causes a loss of function. Pain is adaptive. Okay, pain sends a signal that something is wrong. And you should listen to those particular signals, particularly in the case of injury, and go, oh, wait a minute, maybe I shouldn't be doing that. Okay, so bringing blood flow to the area of injury, which brings immune cells and repair cells to that site to start resolving the problem, and then a temporary loss of function to give you some time of recovery. Okay? So, why do we get redness and heat? What's going to be responsible for that particular symptom? Yeah, bringing more blood flow, which has a lot of red cells to that particular site of area. That's what causes the redness, is more blood flow to that particular area. And the cells in the blood, and the blood is warm, are metabolically active. Increased blood flow brings increased heat to the area. What causes the swelling? Another word for swelling is edema. What causes edema? Pardon me? It's not so much a product of the cells being brought to the area. What happens to tissue when it swells? Fluid accumulates in the tissues. Where is that fluid initially? It's in the vessels, okay? It's part of the blood serum, okay? It's in the circulatory system. So during inflammation, what is it doing? That fluid is leaking out of the circulatory system and into the surrounding tissues and causing swelling or edema. So what's causing that? What's causing that fluid to start leaking out of vessels when it previously doesn't do that? Well, that's going to be an important role that the cytokines will play. They cause changes in the diameter of the blood vessels. Blood vessels can dilate. And it can also cause permeability changes in the walls of those endothelial cells. And so vessels under the influence of certain cytokines get more leaky, and more fluid goes out into the surrounding tissues. And what else does that allow? If those blood vessels get more leaky, it's easier for immune cells to crawl out of the circulatory system and get into the surrounding tissues. What do we call that crawling process that immune cells can do? Diapedesis, okay. So why do we get pain? Well, what happens is that fluid starts to accumulate in the tissues and they swell. There can be increased pressure on nerve endings in those tissues. Also, some of these cytokines irritate nerve endings as well and cause direct effect on the neurons and give us that sensation of pain. So pain can be a chemical phenomena, but it can also be a pressure phenomena of that fluid pressing on cells, particularly neurons, and sending us that pain signal. So here's a slide that shows you a kind of overview of what's happening during the inflammatory process. So let's take a look at this and go through this. Okay, so we've got our damage to the tissue, we've got our splinter. And that splinter probably contains some microorganisms growing on the wood of the splinter. And so basically we're injecting pathogens into that tissue. Now, we've got some immune cells already patrolling around. Remember, we've got those macrophages that are normally part of the RES system. And they're moving around and checking around. And if they detect some PAMPs on the surface of these pathogens, those PAMPs can interact with the toll-like receptors on the surface of the phagocytic cells, and they can start to gobble them up and start to remove them. But they need help. Okay, they want to recruit more help because there might only be a few of these macrophages or neutrophils. We need a lot of them if there are a lot of infectious agents, particularly ones that are multiplying and growing. Okay, every 20 minutes we get a new generation. So these neutrophils, particularly when they ingest things, can degranulate, and those cytoplasmic granules can start releasing their substances, some of which are cytokines, which are inflammatory substances which can have an effect on the vessel walls and change their permeability and change the diameter of the vessel walls. So here's my little model of a vessel wall, my hands right here, okay? When vessels are constricted, the cells are very tightly pressed to one another, and the intercellular spaces between them are almost absent. But what happens if that vessel dilates? It starts to pull apart a little bit. Okay? And the spaces, the intercellular spaces between those cells start to get bigger and bigger. And it gets easier for fluids to leak out and white blood cells to then move out by diapetesis into the surrounding tissue to help reinforce these guys that are here fighting the early stages of the battle. And what's going to happen? Some fluid starts to accumulate here. Tissues start to swell. We get pain. Some of those particular compounds that are produced by the damaged tissue produces some cytokines, and these neutrophils that are phagocytic are also chemotactic substances which attract white blood cells. The more that the blood vessels dilate, the more volume that they can accommodate. So dilation of blood vessels tends to bring blood flow to that particular area. So if we've got a particular area of injury and these vessels locally dilate, that will increase the red number of red cells in that particular area. So we get the heat and we get the redness. Eventually, cell repair is going to start to take place in the walls of this vessel to seal off the injury. There's going to be some clotting mechanisms that are activated. And then these recruited white blood cells are going to hopefully resolve the problem by eliminating the pathogens. The external skin starts to divide and repair itself again. And macrophages can come in and kind of clean up any damaged tissue. And hopefully this will resolve itself completely. And in a relatively short period of time, things are back to normal again. If that's the case, then we're said to have had an acute inflammatory response. It happens, it works, and things are resolved. Where we can get in an acute inflammation is protective, okay? Its purpose is to repair the damage. Where we can get into a real problem is if the inflammatory response doesn't shut off, it keeps going and keeps going and keeps going, and we have a chronic state of inflammation. That can particularly happen in situations where we have autoimmune disease, and we start responding to our own tissues. Rheumatoid arthritis is a really nasty autoimmune disease that causes a chronic state of inflammation. And people's joints become permanently swollen, they're red, loss of function becomes pretty much permanent, and that's really a very debilitating type of disease. And so inflammation out of control can be a disease as opposed to a protector. Now, there are just several other slides in here that show you different diagrams. One of the things that I do want to point out is that there are some very important cytokines which are involved in this inflammatory response. The cytokines tend to fall in three general categories. Those cytokines that cause vasodilation, that cause blood vessels to get bigger around and become more leaky. This includes histamines, serotonin, bradykinin, and prostaglandins. And I want to mention the last two just a little bit more. Bradykinin is produced during clotting reactions. So this is a byproduct of the clotting process. Now remember, the clotting process is primarily a mechanical process to seal off damaged or ruptured vessels. But as those clotting proteins are produced, bradykinins can be produced. And they stimulate other cells, particularly neutrophils and mast cells, to degranulate. And one of the things that they produce when they degranulate are these molecules called prostaglandins. Prostaglandins are very much involved in the inflammatory response. They tend to be very potent vasodilators. We're going to mention prostaglandins again in just a second in another context. Okay, some of the chemical cytokines produced during inflammation are chemotactic factors. So they're responsible for trying to attract white blood cells to that site of infection. And these are an example of some of the things that tend to be chemotactic. Fibrin is a product of the blood clotting process. So it tends to be chemotactic. So when clot forms, that tends to attract white blood cells. So that's pretty good. And then there are some substances that have both vasodilating and chemotactic effects. Some of the complement fragments fall into this category. Some of the interferons. And then there are a variety of interleukins, platelet secretions. Here's another inflammatory mediator you might have heard of before, leukotrienes. They're actually involved in asthma, which is another type of allergic reaction. Asthma is an out of control inflammatory response. So, here's a blood vessel, normal blood vessel. Cells are very tightly pressed to one another. Very minor spaces between the endothelial cells that line that particular vessel. So there's minimal fluid leakage out into the surrounding tissues. And any fluid that does leak into the surrounding tissues eventually gets into the lymphatic system and is returned to the circulatory system. It's pretty difficult for these white blood cells to move through those constricted vessels. But under the response of inflammatory mediators, vasodilation occurs, and that allows white blood cells to move out into the surrounding tissues. It allows more fluid to move into the surrounding tissues. The extra fluid does cause swelling, but it also can dilute any toxins that are present, so there's a diluting factor. And then there's all these blood clotting proteins that are present in the blood serum, and they start leaking into the surrounding tissues as well to facilitate the clotting process. Here's another set of diagrams that just illustrate the same thing. So here's our injury, bacteria invade. Damaged cells start to release cytokines, prostaglandins, leukotrienes, histamines. Okay? And these all have an effect on the blood vessels. Okay? They make the blood vessels more permeable. Histamine makes more vasodilation. That increases our blood flow to the area. Okay? Macrophages and neutrophils start to squeeze out now into the surrounding tissues. And increased permeability allows antimicrobial chemicals and clotting proteins to seep into the tissues. That causes some swelling and pressure on nerve endings. But eventually things start to resolve. You start to form clots. Vessels start to be repaired. Leukocytes, macrophages, neutrophils start to clean up the damage that's occurred. And stem cells start to divide to repair vessel walls, skin fills back in again, and hopefully over a period of a relatively short period of time, inflammation corrects the imbalance that occurred as a result of the injury. This is just a flow diagram. That's another way to organize and show you some of the effects that these various stimuli have. Tissue injury is the stimuli that starts this cascade that ultimately promotes healing. This is just a summary again of some of the types of chemical mediators that are involved that have vasoactive effects, that have chemotactic effects, and some of the cells that produce these particular things. Okay, one other aspect of your immune system that sometimes, but not always, accompanies inflammation is fever. What causes fever? Well, ultimately, what causes fever is that the temperature sensor in your hypothalamus of your brain gets reset. Basically, the thermostat gets turned up. So now the brain is telling the body 98.6 degrees Fahrenheit is not the normal temperature. It might be 102. What causes that reset? Compounds called pyrogens are responsible for resetting your thermostat, basically. Pyrogens are of two types. They're exogenous or endogenous. Exogenous pyrogens are things that stimulate fever that are external in origin, like components of bacterial cells. So lipid A, which is part of the LPS layer, that's an endotoxin, is an exogenous pyrogen. Various immune cell cytokines are endogenous pyrogens, including interferon. Prostaglandin. Remember that was produced during inflammation? Prostaglandin seems to be one of the big players in initiating fever. If prostaglandin starts to be secreted and gets into the hypothalamus, it resets the temperature sensor, raising the bar, and then the body responds because it thinks it's too cold. It's 98.6 degrees Fahrenheit. The thermostat says normal temperature is 102. So what does it do? It initiates mechanisms that generate heat. Shivering, for example. That muscular contraction generates heat. Increased metabolic rate generates heat. Inhibition of sweating, vasoconstriction, which tends to divert blood flow into the core of the body, because core body temperature as opposed to peripheral body temperature is really what's most important in terms of survival of the organism. So what these set of activities do is start raising your body temperature to the new set point that has been established by the hypothalamus. Well, having a fever can be pretty uncomfortable. But there are benefits of fever. Generally speaking, your immune cells are more active at a higher temperature. I mentioned earlier on that they produce more interferon at a higher temperature. And that's why catching a cold could potentially compromise your immune system, because the cells produce less interferon at a colder temperature. Increasing the body temperature is like turning up the incubator. And very potentially, if the pathogen has a very narrow range of temperature tolerance, raising that temperature by a few degrees could be lethal to the pathogen. Fever tends to decrease free iron availability. And iron availability is crucial to pathogen growth. So the net effect of fever, at least to certain levels, is beneficial. Now, if you start getting fevers too high, if you start getting up in that 104 degree range, then what's the danger? Protein denaturation, okay, and cells start to die. So you're running that kind of a fine line. A little bit of extra heat, good. Too much, bad. If your fever gets too high, you might want to take one of the antipyretic drugs, anti-fever drugs. Like aspirin or ibuprofen. How do these particular antipyretic drugs work? Well, they inhibit a particular enzyme. They inhibit an enzyme called the COX-2 enzyme. Cyclooxygenase 2. And this particular enzyme is an important enzyme in the synthesis of prostaglandin. So it inhibits prostaglandin production. Well, why is that important? Because prostaglandin is one of the major cytokines that's causing that reset of the hypothalamus. So it suppresses fever in that particular way. Remember that prostaglandin also can be associated with pain by irritating nerve endings. And so aspirin and ibuprofen are also pain relievers in addition to fever reducers. Both of those particular actions are prostaglandin based upon the ability of these particular drugs to inhibit those COX-2 enzymes. So next time you have a fever, you might consider whether you want to take aspirin or ibuprofen. Yeah, it'll probably make you feel a little bit better, but you might be compromising one of the important parts of your body's second line of defense that really helps to augment some of the other aspects of your immunity, particularly those involved in white blood cells and cytokines. Now, you've got to monitor it, and if it gets too high, then that's a problem. And you've got to start treating it. You've got to give people ice baths. You've got to get them cooled down if that temperature gets past that critical point. But up to a certain point, it's really beneficial, even though it's uncomfortable. Okay, so that will be the ending point as far as exam material is concerned. So there are 2 sets of materials posted on Moodle. PowerPoint, lecture notes, study guides that deal with the host-parasite relationship. That's the first one. And then line 1 and line 2 of defense. Okay? All right. So what are we going to do in lab tomorrow? Well, one of the things we're going to do is we're going to start our last project other than our unknown. And this is another medical microbiology project. And what this particular project is going to involve, and you're going to work in pairs on this, is you're going to be given 2 simulated samples. One a simulated urine sample and one a simulated fecal sample. And ask you to identify within some certain boundaries what organisms are there in those samples. Now we're going to pare it down for you and say there's a list of candidates of organisms that this could be. So it's not just anything. And you're going to be asked to determine what's in your stimulated food sample. It's a food sample and a urine sample, not a fecal sample. What's in those 2 particular samples. To do that, we're going to use, in addition to some tests we've already done, like the urease test and the citrate test, we're going to use some differential medium that allow you to start figuring out differences in microorganisms based upon how they grow and what their appearance is on some augurs we haven't used before. So you're going to be introduced to eosin methylene blue, or EMB augur. You're going to be introduced to Salmonella shigella, or SS augur. You're going to be introduced to 2 or 3 other ones. One of the reasons we're doing this exercise is to give you some practice with using keys and flowcharts, because you're going to be using keys and flowcharts in your identification of your unknown. What I'd like to do for the next few minutes is run through a few things that I probably take for granted, but shouldn't. And that is that, number 1, you know how to use a key, for example. And not all of you have done that before. So I've just reproduced the key that we have in the lab manual. This is chart C1. I apologize, it's cut off a little bit. There's a couple of things you're going to have to write in there. And I've got chart C1 up here on the overhead so you can kind of see where you need to write in. This is where you start identifying your unknown. You should not begin to try to identify your unknown without starting at this particular key. This is a key to genus. When you do your unknown identification, first you have to figure out what the genus is, and then you will have to use Bergey's manual to determine what the species is. Notice that we say that in this particular chart C1, that your unknown genus is guaranteed to be in this key. In the back of your lab manual are some other genus keys as well. And they are intended to provide backups, other ways to verify your unknown. But your unknown is not necessarily guaranteed to be in those other keys. So you could go through that key and not find the name of your unknown that you found in this key. That's okay. So on your sheet that I gave you right here, there's probably a couple of things you should do. And the first thing you should do is, up at the very top where it says gram plus, notice that there's a 1A next to that gram plus. You should put 1A beside that, because that got cut off. If you turn your key over, you'll notice that there's a 1B on the other side, and that says gram negative. So when you're doing a key, where do you always start? Might sound kind of trite at the beginning. And typically it is a numbered key, and so you take the first number in the key, and there should always be a paired number with it. This particular type of key is called a dichotomous key. Dichotomous keys give you pairs of choices, and you are asked to look at what you're trying to identify and see which of those choices applies to your organism. Once you make that choice, that then defines where you go next in the key. So for example, if your unknown is gram positive, okay, you're starting out at 1A. The first thing that you're going to evaluate in this key is the gram reaction of your unknown. And so it's a gram positive. You verified that with me. Where do you go next? You go to the choice directly below the one that you selected. That may or may not be the next consecutive number. So for example, if you are gram negative, where do you go? Your next choice is 10, the oxidase test. But notice that there will always be two 10s in the key. There's always a dichotomy based upon it's this or it's this. There's always that fork in the road, and you constantly have to make choices, which direction you go, and then that leads you to the next step in the key. Let's go back to the first page. So if you are gram positive, you chose 1A. The next set of choices you have to determine have to do with maltose fermentation. Did you produce acid from maltose, or did you not? Where do we go if you are positive for acid from maltose fermentation? You start evaluating next 6 because that's the choices below the one that you took. So if you are gram positive and a maltose fermenter, then you have to determine if you are a cocci or a bacillus. What if you're a bacillus? Where do you go? Number 8, right? Directly below the one you chose. Now you are down to a point where if you are a organism that hydrolyzes gelatin, that puts you in the genus Bacillus. If you are not a gelatin hydrolyzer, that puts you in Lactobacillus. So basically, you start at the beginning, make your proper choices, and then follow the proper choice you made until you come to the name of a genus. Turn your page over. So if you are gram negative oxidase negative, where do you go next? What test result do you need? Number 12, right? Because you chose oxidase negative, so you go next choice, directly below the one I took, 12. Phenylalanine positive or phenylalanine negative. If you're phenylalanine negative and indole negative, where do you go? 16, right? What if you're methyl red positive? Then you go to 18, correct? And what if you don't reduce nitrate to nitrite? Then you've got your genus, Klebsiella. This is really a pared down key, and it only contains certain genera, only ones that typically we will use in lab, and all of the unknowns are guaranteed to be one of these particular genera. Now, if you look at the second key that's in your lab manual, it's got a different organization. It starts out with a different set of questions. I'm trying to remember what the... Look here real quick and see. Chart C2. First choice here are cocci and bacillus. So again, you have to make your choice. Now, this particular key is set up a little bit differently. It's set up in such a way that if you choose cocci, it gives you a number over here, and then that's the number you go to. If you choose bacillus, it shows you a number over here, and that's the next number you go to. So the way keys are set up are a little bit different depending upon who's doing the key. Now, there's another type of dichotomous identification tool that you'll find several examples of in your lab manual, and one of them you've already used. And that is a flow chart. So we've got, we've already used that flow chart for a gram-positive bacteria, remember? The keys that we've been talking about so far have numbers on them. The flow charts typically don't have numbers. They just have these T-splits built into the flow chart. But the idea is the same. You start at the top and you work your way to the bottom. This is one of the ones I just pulled from one of the appendices of your lab manual. So this is a flow chart identification scheme for selected gram-negative bacteria. So what do? You start at the top. And what's the first determination you have to make? Is your cell a rod or is it a coccyte? If it's a coccyte, then you take that branch of the flow chart. If it's a rod, you take the other branch of the flow chart. Let's say it's a coccyte. What's the next test? The next test that they do then is an oxidase test. You'll notice that there are two splits there. R stands for result. So oxidase positive on the left branch, oxidase negative on the right branch. Now, you'll notice on the right branch, oxidase negative, it brings you down to a particular species already. You've identified it. Okay? If it's oxidase positive, then you've got a couple of different possibilities. It could be Brandhamella, or it could be various Neisseria species. So there are some remaining branches that allow you to identify those Neisseria species. Here, M stands for media. So the next media that we're going to use is carbohydrate media. And that carbohydrate media is of three types: glucose, lactose, and maltose. You'll notice that the branches, the one on the left is cut off. The three branches give you different results. Plus, plus, plus in the middle one, plus, plus, minus in the next one. So what that would say in the middle one would be glucose was plus, lactose was positive, and maltose was positive. In the one on the right, glucose was positive, lactose was positive, maltose was negative. Okay, so you see how you read these? But you just keep working yourself down until you get to an organism or a group of organisms. Sometimes the key is made in such a way that it can't give you a definitive answer. It will say it's either this or this. I can't tell you. Yep.
All three of them? Or is it positive, negative, or negative?
Well, we don't. Okay, don't even worry about that one because we don't. That's not an organism we even deal with. That one on the left on that bottom chart. So this one over here. It's positive, negative, negative, but don't worry about it because we don't have any Brandhamella species. Now, one of the things you have to be aware of is that flow charts are only good for the organisms that are included within them. So this flow chart only allows you to identify certain organisms, okay? Pseudomonas fluorescens, Pseudomonas aeruginosa, Alcelgeans denitrificans, Proteus vulgaris, Proteus mirabilis. So when you build these flow charts, you have to say, this is the group of organisms I'm building the flow chart for. The more organisms you include, the bigger and more complicated the flow chart gets. So that's why they said this was for selected gram-negative bacteria. Somebody had a need to differentiate between these particular species and made this flow chart to easily allow you to differentiate between those particular species. Okay, that's going to have relevance for lab because in your simulated samples, I'm going to say your simulated samples both are one of the following, and there's a list of like 10 organisms. I'm going to let you use the flow chart that's already been made for gram-positives. So if you get your food and urine sample and they're both gram-positive, then you can just use that gram-positive flow chart and identify them. However, if one of them or both of them are gram-negative, what do you use as a flow chart? You have to make your own flow chart. How do you do that? Well, this is something that I'm going to have you work on at the beginning of lab tomorrow. Practice making flowcharts before you make your own. What do you need to make a flowchart? You need to have a list of the organisms. Oh, I'm sorry, there's only one per group. So share these per group. You're going to need to know the list of organisms that are supposed to be in your flowchart. That's in your lab data. Okay, they'll pass it down to you. And then you're going to need to know test results for those organisms. For what tests? Well, I'm going to tell you which tests you're going to have available to do in this identification. So one of the tests will be a citrate test. Another one will be a urease test. Another will be an oxidase test. So you'll have a list of tests. So one of the things you're going to have to figure out is, what are the results of your candidate organisms for each of those tests? You're going to have to make a little spreadsheet, basically. Where do you find that information? By digging through Bergey's manual. You're going to have to look these things up and say, is this organism citrate positive or negative? Is this organism urease positive or urease negative? So what I've done in this little exercise that I handed out is I've given you a hypothetical set of data. Like that you might come up with. And I've just randomly chosen some metabolic tests. Some we'll use in our exercise, some we won't. And then instead of giving species names, I've just said species A, B, C, D. Okay? And then I've just randomly made up results for those species. So if you look at your test results here, species A is a gram-positive rod. It doesn't ferment lactose, but it ferments glucose. It's oxidase negative, it's citrate positive, etc. Species B is a gram-negative rod, lactose positive, glucose positive, etc. So what I want you to do is I want you to make a flow chart. And instead of having, Proteus vulgaris or Proteus mirabilis down at the bottom, you would have species A or species B or species F, okay? And then the flow chart would be how you get there. Everybody's flow chart should be different because it just depends upon what characteristic you want to start with. Some of you might decide, I want the first decision to be citrate positive or citrate negative. Others might decide, I want it to be gram positive or gram negative. If all the organisms have the same result for a test, should you include that test in your flowchart? No, it doesn't give you any information. It doesn't eliminate anything. Because remember what happens in flowcharts as you work through there, choices you make start to split organisms into groups. Okay? But I think it's really an important exercise to go through constructing a flowchart like this, because once you learn how flowcharts work, it's much easier to deal with the complexity of flowcharts that have a lot of species in them. You understand how they're constructed. Okay? So we'll do this little exercise at the end and just kind of see what flowcharts you come up with. But then ultimately you will be given this list of organisms and you'll have to make a gram-negative flowchart in case you have a gram-negative unknown. I'm going to look and see, but I think I'm probably going to have you inoculate your unknown on some of the media that we're using for this exercise as well. So we'll probably have you do an EMB plate and I forget what the other plates are. So I'll give you some instructions about that tomorrow. Okay?